Hello students. So by now you have learnt all the events that are essential or that are part of the pre-fertilization processes. And in these events you have learnt how the male and female gametes are developed. And in the later parts you have learnt about pollination, and you have learnt that the male gametes are going to come and sit on the stigma. And you have also learned that a variety of the pollen grains actually come and sit on the stigma, and it is not only one type. And of these, it is only the compatible pollen grains, right, that is actually allowed to germinate. So when we talk about pollen germination, we talk about the pollen tube growth. And when the pollen tube is going to grow, the pollen tube reaches the female gametophyte, which is nothing but the embryo sac. And you have already learned that the embryo sac is nothing but a 7-celled but 8 nuclei condition. Now 8 nuclei because in the central cell you are going to have not one nuclei, but you are going to have two different nuclei. So as of now, I'm sure that you already know that when the pollen tube reaches the female gametophyte or the embryo sac, it is going to release two male gametes. It is not one, but it is two male gametes. We are talking about male gamete nucleus, or we can also use the word sperm cells. So there are going to be two sperm cells that are going to be released inside the embryo sac when the pollen tube is coming in contact with the synergids. Now the question is, what's going to happen with the two male gametes? Because we know fertilization is the event of the fusion of male and female gametes. If we are going to have the fusion of male and female gamete, we require only one male gamete that is actually fusing with one female gamete, and that is actually referred to as the fertilization. One male gamete and one female gamete, right? That is going to give rise to the process of fertilization. Here the obvious question is why there is a requirement of two different male gametes or two different sperm cells. Now to start with, you have to understand that here the process is slightly unique. Okay, why do I say it is unique? Because in here the process is referred to as double fertilization. So the obvious question is, what do you mean by double fertilization? So in simplest term, if there are two fertilization events that are occurring at the same time, it is referred to as double fertilization. So why it is important and how it is unique to this angiospermic plants or the flowering plants, let's have a look. So to start with, you all know that there are two types of cells in the pollen grain, and one of them is going to be the generative cell. The other one is the vegetative cells. But the generative cell, as you know, give rise to two male gametes, and these two male gametes are going to participate in two different events. Let's see what are these. The first one is one of the male gamete is going to fuse with the egg cell. Now egg cell is going to be haploid in nature, and when we say haploid, we are talking about only one set of chromosome is going to be present. And when they fuse, the egg cell and the male gamete fuse, one of the male gamete fuse, they'll give rise to the zygote. And remember, this zygote is going to be diploid. Okay, and this is again a unicellular structure. It is made up of a single cell. Now the zygote is diploid, means it is going to have two set of chromosomes. So there are going to be N and 2N. N is actually haploid and 2N is actually diploid. Now when we say haploid we say we see in case of gametes, both in case of the plants and animals, the gametes are typically haploid. Now the remaining cell, that is the somatic cell, we typically refer to them to as the somatic cells, and the somatic cells are having two sets of chromosome and they are diploid. And whenever the gametes are going to form, they are actually formed from the diploid somatic cells, and the process we all know involved is going to be the meiosis. Okay, that's all we need to understand regarding the haploid and diploid. But again, going back to the process of formation of zygote, again it is a process of fertilization, because we are involving one male and one female gamete, and this event is called syngamy. On the other hand, the remaining male gametes, right? As we said, there are two, so the remaining male gamete is going to fuse with the central cell. Now the central cell is going to have two polar nuclei, and these two polar nuclei are going to be haploid in nature. So here we are going to have one nuclei here, second nuclei here, and third nuclei here. So here three different nuclei are fusing to give rise to PN, that is going to be triploid in nature. So what does this PN stand for? PN stand for primary endosperm nuclei. As we are going to have the involvement of three different nuclei in this fertilization event. Remember, this is also a fertilization event. Why? Because here also there is fusion of male and female gametes. Male gamete is of course the sperm cells, and the female gametes is represented by the central cells, two polar nuclei. As we are going to have the involvement of three different nuclei in this fertilization event, it is referred to as triple fusion. Now if you can look here, there are syngamy, it is a type of fertilization, and we are going to have triple fusion, which is also a type of fertilization. So here there are two different type of fertilization happening or occurring at the same time, and that is why this process here is referred to as double fertilization. Remember, double fertilization refers to two different fertilization events of syngamy and triple fusion occurring simultaneously in case of the flowering plants. Now why is it important? Because remember, the zygote is important because in future, in the further developmental events, it is going to give rise to the embryo, and you all know that embryo is a very important structure post fertilization. On the other hand, this primary endosperm nuclei give rise to PEC. Now what PEC stands for? PEC stands for primary endosperm cell, and this primary endosperm cell or the PEC give rise to a very important component that is referred to as the endosperm, and it is important because this in future is going to give rise to the nourishment or the nutrient to the developing zygote that will develop into the embryo. So this is what you have to understand with regards to the double fertilization events. After fertilization, there are certain changes in the embryo sac, and one of the primary change you can see directly in the center. You can see here that here we had the central cell, and the central cell is now represented or it is developed into the primary endosperm cell. And if you remember, the polar nuclei that we had is actually developed into the primary endosperm nucleus, that is again triploid in nature. Now again you can see here there are degenerating antipodal cells. Okay, let us actually write down the different ends. So here we are going to have the micropilar end, and in this side we are going to have the chalazal end. Okay, so on the micropilar end, if you can see the synergids, right? Synergids main role was to allow the pollen tube to actually gain entry to the embryo sac, and as the purpose is served, there is actually no more requirement of the synergids, and that's why they will start degrading. Here both of these synergids are going to degrade. Now of course the egg cell that we had, the egg cell will develop into the zygote post fertilization, and on this side the antipodals, right, on the chalazal end, the antipodals, right, three of them, and these three also will start degenerating. So as you can see, there are these changes that are going to be seen after fertilization in the embryo sac. So that's all for this video. Thank you all for watching this.